today we will hear another one. And to introduce the speaker will be Professor Chongli Yuan, but I wanted to say a few words before we begin with the seminar. And it is with some great personal feelings that I want to describe something that has happened just yesterday. Professor Neil Amundson, who is many times considered to be an architect of modern chemical engineering, died yesterday at age 95. He was a remarkable person. To call him as a giant of chemical engineering would be an understatement even. He revolutionized chemical engineering. He brought new directions, which included essentially changing what at that time in the early 50s was a qualitative description of chemical processes into a more mathematical analytical basis, and created in some ways the word science in chemical engineering science, when we say. So this idea came from Professor Amundson through his own personal work, and remarkably, in addition to that, he also created a whole department of chemical engineering at the University of Minnesota, which was in obscurity when he became head in 1949, and then he remained as head for 25 years, and in this period brought the school from essential obscurity to a #1 position in the rankings of all sorts, and very high ranking that it still holds even today. And in some of those periods, we used to say there is no #2 because the #2 was so far behind. He had a remarkably fruitful career. He recruited outstanding faculty, of course, by which that department became what it did. And I want to say a few words about the strategy that he employed for a very young person. He was 35 when he became the head of the department. He hired faculty when I was a student there myself. I myself am a Neil Amundson student, so I say this with great feeling myself. There were 15 faculty, but 6 of them were non-chemical engineers. There were a microbiologist whom he hired on the faculty, Henry Surya, in 1957, a mathematician, Rutherford Harris, whom he hired in 1958, and others including Ted Davis, Stanley Schmidt, Bob Carr, and John Dollar, all physical chemists. So a faculty of 15 had 6 non-chemical engineering faculty. We're talking about in the 60s, 70s, 80s, until now. So we talk about interdisciplinary work today, but this was an individual who saw this 50 years ago and created by his vision this idea of multidisciplinary research that we now talk about so much. And this was going on there, and that was the key for the success and the growth of that school. There are many faculty members in our school who are graduates of the University of Minnesota and have had a direct contact with Professor Amundson and others. I myself am a direct PhD student of Neil Amundson, and I believe I'm the only one in this department who is. The other faculty member who is also very close to Professor Amundson is Professor Ramakrishna. Although he was not a direct student, they wrote many papers together after he finished his PhD, in addition also writing a textbook, which many of you may have been using when he teaches the course. So we salute this giant of chemical engineering who changed the direction of chemical engineering, and we are so fortunate that we have still many of his directions being followed, and many of his representatives are here in our own school. I request all of us to have a 30-second moments of silence in memory of Professor Neil Amundson. Thank you. Please introduce the speaker.
So it's my great pleasure to introduce Dr. Surya Mallapragada today as our Centennial seminar speaker. So she used to be like one of us. So Surya started her education in chemical engineering from IIT Bombay. And she graduated in 1993 and decided to choose Purdue University to pursue her PhD degree. It took her 3 years to finish the PhD degree, which is quite a record. And afterwards, she decided to start her appointment at Iowa State University. So currently, she's the department chair of chemical and biological engineering and the Stanley chair of interdisciplinary engineering at Iowa State University. She also holds courtesy appointments in the material science and engineering, as well as neuroscience department at Iowa State. So Dr. Mallapragada has also served as a senior scientist and program director at AMS laboratory, which is a USDOE laboratory, and her research interest spans from polymeric biomaterial to tissue engineering. She's particularly interested in drug and gene delivery, as well as bioinspired materials. So she has received many awards in her career, among which I'm only going to mention a few. She has received the prestigious NSF Career Award, a 3M non-tenured faculty award, and she has also been named as one of the top 100 young innovators by MIT's Technology Review magazine. So she's also fellow of AIMBE and AAS societies. So without further introduction, I'll give the stage to Dr. Mallapragada and look forward to a great talk today.
Thank you so much. Can everybody hear me? Thanks a lot, Dr. Giron, for the very nice introduction.
We need the lights on because we are
Okay. So it's wonderful for me to be here, especially for this momentous celebration for the Centennial, and it's an honor to be picked as one of the Centennial speakers. So I'd like to thank the department for selecting me to essentially represent one of the Centennial speakers. And even though I was here for only 3 years, it was a very key time in my life because it really laid the foundation for my research career. Not to mention that I also met my significant other here at Purdue, so it's really changed my life. And some things never change. I remember being on the other side of this room for seminars, and even at that time there was nobody in the front row. So some things just stay the same, continuity, I guess. I had initially given a title about bio-inspired materials, but I thought since we have a diverse audience, I really like to talk about a couple of different projects, so that way there'll be something of interest for hopefully everybody. And so I'm going to talk a little bit about gene delivery and also about biomineralization with the bio-inspired materials. So this is a photograph of Sweeney Hall, which is the home of the Chemical and Biological Engineering Department at Iowa State, and just across the street we have a new Biorenewables Research Lab, which is the home of the new NSF Engineering Research Center on chemicals from biorenewables, and we have Ames Lab, which is a Department of Energy lab situated on the Iowa State University campus and focused on materials research. So it's been a great resource for my own research career. I have an appointment in Ames Lab and take advantage of the national lab facilities on campus. Well, I'm going to start off with acknowledgement just so I make sure I don't miss out anybody at the end if I'm running out of time. All of the students who are involved in the work, my collaborators, most of my projects are collaborative, and so both my collaborators at Iowa State and elsewhere, and of course the funding agencies that made this work possible, without which this would not have happened. So what I'm going to do right now is to just talk about the broad area of designing, synthesizing, and characterizing a family of block copolymers in our lab that we've been interested in, and talk about two different applications. The first one dealing more with the gene delivery aspects, and the second one involved with biomineralization. And it's the same family of copolymers that we're looking at, and so that's the thread that's tying this together. You can wait till the end for questions, or feel free to interrupt me at any time. I'd be happy to answer questions as we go along. So some of the materials that we synthesized a few years ago in our lab are these pentablock copolymers, which have a central block, which is made of a triblock copolymer of polyethylene and polypropylene oxide. And then we have two end blocks, which are essentially polyelectrolytes. In this case, I've shown polydiethylaminoethyl methacrylate. And so they have some interesting properties. What we were going for was to make these amphiphilic block copolymers, these central blocks themselves, as you change temperature and concentration in aqueous solutions, end up forming micelles. And as you increase the temperature and concentration further, they end up forming physical gels. And that's been known for a while. And the idea of attaching or having these additional blocks, which are polyelectrolytes, really gives us another handle in that we can vary pH and now have different sorts of behaviors, such as the gel dissolving fast or slow, depending upon changes in pH in addition to the temperature effect. So our motivation really for doing this was to look at these as self-assembling templates for biomineralization to form injectable nanocomposites, because these materials at low temperature in aqueous solution, for instance, are liquid, and then you can inject them, say, subcutaneously, and at body temperatures they form a physical gel. And then that can dissolve slowly to produce either modulated drug release or, in the case that I'm going to talk about in a little more detail, for sustained gene delivery. At the same time, these can act as templates for nucleation and growth of inorganic nanocrystals. So you can essentially have injectable nanocomposites, which could be useful for a variety of different applications. And so we've synthesized these polymers and shown that they go from unimers to micelles in solution to physical gels that you can see here. It's a self-supporting gel, and this transition can occur at reasonable concentrations between 20 and 25.8% in water and at temperatures. So if, say, at room temperature you end up with either unimers or micelles, and at 37 degrees centigrade you end up with the gels. Interestingly, we've also seen that they not only form gels but also elastic solids, which are have lower water content. This is actually an example of a self-assembled cylinder solid, elastic solid, which has very low water content. The gels typically have 80% water content or so, whereas these have more like 28% water. But this is formed completely by self-assembly and exclusion of water. This happens at a slightly higher pH. If you recall, we also have the pH sensitivity with these polymers, and as you increase the pH, it becomes much more hydrophobic and excludes the water. And the PDAM also exhibits what's called LCST behavior, and so we end up having a much more dehydrated gel or a solid. And we've done some small angle X-ray scattering of this solid and shown that it has a very organized structure. So at these higher pH values, instead of spherical micelles, you end up forming cylindrical micelles, which then self-assemble into a hexagonal close-packed structure to form this organized elastic solid. And so we can get much better mechanical properties with these elastic solids compared to just the physical gels. If you look at just some DMA with temperature, you can see that if you take this bulk copolymer and precipitate it in an organic solvent, you get very different mechanical properties as opposed to precipitating it at high pH in an aqueous solution where you form this self-assembled solid, and then it has much better thermal stability over a much wider range of temperatures. And so we've shown that we can form these or synthesize these block copolymers, and in fact we've actually synthesized a family of block copolymers. I've just shown you as an illustration the one with the polydiethylaminoethyl methacrylate, but we've actually made a family of different copolymers with different polyelectrolyte end blocks, which can exhibit similar behavior in terms of being able to respond to temperature as well as pH. And I'm going to talk about the first application, which is in gene delivery. So the gene delivery process is mediated by polymers. So we have here a schematic of a cell with a nucleus, and we have a polyplex, which is a polymer DNA complex, which has to be internalized into these cells and has to escape from this low pH environment, which is the endosome or endolysosome, and protect the DNA from being degraded in the process, and also get it to the nucleus so it could be expressed in order to facilitate efficient transfection. So these are some of the intracellular barriers to delivery. And of course, viruses do a great job with gene delivery because they're very efficient doing this. But as far as polymers are concerned, they're not terribly efficient, partly because of the need to overcome a lot of these intracellular barriers. So with these pentablock copolymers, especially the cationic ones, such as the PDAM-based ones that I just showed you, they have some advantages or desirable properties in that they do form micelles, which is good for cellular entry, and also show these thermoreversible gelation properties, which allow them to be injected even intratumorally if the tumors are accessible. We're looking at gene delivery for cancer therapies, suicide gene delivery for cancers. And the biocompatibility issue with viruses or viral vectors, you have a lot of immunogenicity issues sometimes. Of course, because of the cationic nature, they can exhibit complexation with DNA, and the sustained delivery because of the gelation process can allow us to take advantage of long-term delivery over time, even though the expression is just transient with these polyplexes. So we have here just some images of polyplexes. This ratio shows the ratio of the amine groups in the polymer to the phosphates in the DNA, which is essentially some molar ratio of the polymer to DNA. And we have here one of the big issues with cationic polymers, such as XGen, for instance, which is linear polyethylene imine, which has been used or researched extensively for gene delivery. It's one of the gold standards. Has a lot of toxicity issues. Lots of cationic groups, which not terribly great if you do a LDH assay to look at cytotoxicity. We see that with these pentablock copolymers, they're much more biocompatible, much less cytotoxic, because you don't have just the cationic groups, you have all of these non-cationic groups as well, which help with some shielding of these cationic groups, excess cationic groups, and which can contribute to this cytotoxicity issue. The next thing we wanted to look at is whether these polymer DNA complexes, DNA is negatively charged, so it's an electrostatic interaction with these positively charged polymers, but can these polymer DNA complexes form gels just like the polymer can? And sure enough, the answer is yes. And not only do they form gels, but when these gels dissolve, what you end up seeing is a release of not just free DNA or free polymer, but release of polymer DNA complexes with time, so we can get sustained delivery because these polyplexes are being released, and we've also verified that with this gel electrophoresis. So what we have is a system which could be used for sustained delivery, which can release these polyplexes over time. Of course, then we looked at transfection in cancer cells. These are human ovarian carcinoma cell lines, and used a couple of reporter genes. This one is with the green fluorescent protein. We have comparable transfection efficiencies as XGen, and whether we look at percentage of positive cells or if we look at actual protein expressed. And we've also done a few in vivo studies. These are subcutaneous tumors on nude mice, and you can see These, we have very localized delivery. The fluorescence is mostly in these tumors as opposed to other areas. So these are pretty good vectors for cancer, cancer therapies. One of the interesting things that we were not expecting that we did see in our studies was selectivity. So when we looked at transfection, we focused mainly on cancer cells, and we had pretty good transfection efficiencies, as I showed you. But when we looked at normal cell lines or non-cancer cell lines, we end up seeing very little transfection. So what we did was to do co-cultures of, say, normal human these are cancer cells, HD1080, which are stably transfected to express this red color. And the reporter gene was used in transfection with this so that we could co-culture cells, normal cells and cancer cells, and we can see selectivity of transfection. And this is what we see. With these pentablock copolymers, this is a cancer cell line. We see pretty good transfection efficiency compared to normal cells, which is essentially close to 0. If we take a vector such as XGen, which is linear polyethylene imine, we have comparable transfection in human cells as in the cancer cells as well as the non-cancer cells. And this is true whether we culture them separately or in co-cultures at different ratios, we see similar results. So this is something that is good for us because we want to have selectivity of transfection in cancer cells as opposed to other cell types, but we were not sure as to why this was happening. And so one of the things we wanted to do was to dig deeper into this process to look at the reasons for this selectivity of transfection. And to do this, we looked at a couple of different methods to do this. We attached EGF, and this is a busy slide, so I don't want you to look at each of these graphs, but just the main take-home message. We have a couple of cancer cell lines and a couple of normal cell lines, which are non-cancerous. And what we have here is attachment of EGF, which is epidermal growth factor, which has been known to target cancer cells. Attachment of EGF has been used in many studies to target cancer cells because many cancer cells have overexpression of EGF receptors on their cell surface. Another thing that we did was use chloroquine, which is an agent that's known to promote endosomal escape. Chloroquine is very toxic, so it's not something you'd actually use in a therapeutic or in vivo application. But here we were just using the chloroquine to understand the mechanism behind the results that we just saw. And the third thing that we did was attach nuclear localization sequences to the polymers to promote nuclear entry. So the whole purpose of this was to see what the intracellular barriers to transfection was, whether it was cellular entry, whether it was nuclear entry, or whether it was escaping from the endosomes. And when we did this, we did not see much of an effect with either the attachment of the NLS sequences or with the attachment of the EGF, which gave us an indication that those two were not the main intracellular barriers. But when we added chloroquine, these bars and the white on top show you the additional transfection that we got upon addition of chloroquine. So you can see that in the cancer cell lines, there wasn't a huge difference. There was still a big difference with and without chloroquine, but not as big as what we see in the normal cell lines. So what this really tells us is that essentially the addition of chloroquine increased gene expression up to 200-fold higher. This is a log plot here, I'll point out. 200-fold higher in normal cells than in cancer cells. And that tells you that without the chloroquine, the normal cells are not facilitating endosomal escape. So the polymer is not facilitating endosomal escape in normal cells. So why is that? There have been some literature studies in the past showing that the lysosomal pH of cancer cells is actually higher than that of non-cancer cells. And this in turn leads to different extent of damage to the DNA. And we can see that if we just do a simple study looking at the effect of pH, we see a lot more degradation of the DNA at low pH values. And so what's happening in these normal cell lines is that the pH is much lower in the endolysosome, and the polymer can do a much better job of facilitating endosomal escape in the cancer cells as opposed to the non-cancer cells, which in turn gives rise to the selectivity of transfection. And so we've shown that in this part of the work, we've shown that we have been able to synthesize injectable sustained gene delivery systems which can deliver genes over time, which can then permit localized delivery to tumors and have pretty good transfection efficiency comparable to XGen without the cytotoxicity associated with XGen. And in addition, we can provide selectivity of transfection. And this is actually, most of the targeting studies in literature have focused on attaching targeting ligands to target specific cell types. But what we are doing here is using a new paradigm for designing targeting based on the chemistry of the vector, taking into account intracellular differences between cell types, not just looking at differences on the cell surface and looking at cellular entry. So one thing that I did not talk about is that we also developed a new method to investigate polyplex unpackaging based on quenching of quantum dots. One of the things that also needs to happen once these polymer DNA complexes get into the cells is unpackaging so that they can be expressed. And this method actually allows us to investigate the unpackaging mechanism inside the cells. So that's one of the stories I wanted to talk about. The second story is looking at bioinspired materials and biomineralization using the same polymer templates that we just talked about, two different applications with the same set of polymers. So our motivation really is to try to mimic self-assembly that happens in nature. Essentially looking at a bottom-up approach to materials design instead of a top-down approach. This is a very wonderful example of a glass sea sponge skeleton that Joanna Eisenberg and her co-workers have looked at, which shows so many different structures at these different length scales, whether you look at 1 centimeter or 5 millimeters or microns or nanometer. And it's really the structure that leads to the wonderful properties that this exhibits. And similarly, we have other examples like bone and so on. So what we wanted to do is to take advantage of these self-assembling polymers. They form micelles, and then these micelles form these gels or solids. Now, in addition to these polymers, we wanted to attach mineralization proteins or peptides to the ends of the polymer chains that promote selective nucleation and growth of an inorganic phase, and then the attachment of the growth of the inorganic nanoparticles at these locations can then be used to control their placement in a larger polymer matrix. And so this is shown here. We have these micelles. You have this coating of the inorganic phase, which then self-assembles to form in an organized way, maybe a BCC, FCC, or HCPC structure, form these nanocomposites. So of course, the trick here is to make sure that we just don't precipitate the inorganic without control. We need to have nanocrystals form, and we need to be able to control their location. So in addition to the PDAM block copolymer templates, we also looked at zwitterionic. So in addition to the cationic, we also did synthesize some zwitterionic as well as anionic block copolymers. This was because there have been some studies that have shown that, for instance, calcium phosphate precipitation is facilitated by these carboxylic acid groups. So we've looked at a number of different templates, and I'm showing you a couple of different ones here. And one of our first studies was with hydroxyapatite or calcium phosphate based nanocomposites. So the polymer was conjugated with hydroxyapatite nucleating peptide, which served as the mineralization agent. And we looked at a bunch of these different templates, whether it's pluronic or pluronic attached to this peptide, or the penta-PA, which is the anionic template, or the zwitterionic one. And we can see we've also looked at, in collaboration with Tim Deming, we've also looked at block copolypeptide templates, and I'll talk about why that's important. But what we see in the lower part here are these nanocrystals of calcium phosphate or hydroxyapatite that are formed in these systems. So in this case, these are TEM images of these nanoparticles after we've removed the organic so that we can actually look at these clearly. With TEM, you can see these needle-shaped nanocrystals. So we're not just precipitating the hydroxyapatite in large chunks. We're actually forming these nanocrystals. In fact, they have morphologies similar to that seen in bone. And if you just look at a bulk sample, this is what it looks like. So why is this important? These are injectable nanocomposites, and so you can think about different applications. In fact, we have a collaboration with some orthopedic surgeons at the University of Iowa, where they're looking at post-traumatic osteoarthritis or prevention of post-traumatic osteoarthritis. So after trauma, they want a material that they can just inject into the cartilage to stabilize the cartilage, which has properties, mechanical properties similar to that of cartilage, and can fill the cracks so that you don't have further damage and chondrocyte death. By having this injectable nanocomposites, you can start off with the precursors and your block copolymer templates, and once it actually hits body temperatures in the joint, that's when it forms this nanocomposite. And so you have a way to actually fill the cracks in the cartilage without forming a nanocomposite ahead of time. And of course, then you can't control its shape. So we think it's the first injectable nanocomposite that we have actually come across in literature. And again, our ability to make a nanocomposite with an organized structure, and we've looked at the structure of these nanocomposites with both small angle neutron scattering and small angle X-ray scattering, because it gives us very complementary information. The small angle X-ray scattering gives us more information about the inorganic phase, while the small angle neutron scattering gives us more information about the polymer templates. And you can see that even in the presence of the peptide, or these are the mineralized templates, we still see good organization or similar organization of the polymers in these matrices. And so you don't have disruption of the order in the presence of the inorganic phase. And you see increased scattering when you have mineralization, which is what you would expect in a small angle X-ray scattering, where the scattering is mainly from the inorganic phase as opposed to the organic phase that you can see from sands. So we still have these organized structures that lead to the formation of these nanocomposites. And in addition, my collaborator Klaus Schmidt-Rohr in the chemistry department at Iowa State has also done solid state NMR to prove nanocomposite formation in the bulk by looking at the spin diffusion studies, where you look at transfer of magnetization from the polymer to the inorganic phase, and based on the time that it takes, you can estimate a length scale. And in this case, the length scale that they've estimated is of the order of 18 to 20 nanometers, which matches really well with what we see in the TEM, though TEM will only give us a snapshot in time. This is more of a bulk measurement, which tells us that throughout this, this bulk material, we do have a nanocomposite with just these 20 nanometer needle-shaped crystals in, with a tight interface with the polymer. So we see different kinds of ordering depending on the different templates that we use. And most of the phases that we have are hydroxyapatite. In one case, we have bruchite, which is another form of calcium phosphate, but we can form these nanocomposites, and these are now being tested in joints for post-traumatic osteoarthritis. Another example of biomineralization that we're also looking at, or we've looked at, is looking at magnetotactic bacteria as inspiration for our materials. These are bacteria which have chains of magnetic nanocrystals in them. This is a close-up of these chains. These are magnetite, Fe3O4. And they have these proteins that are an integral part of this magnetosome membrane around each of these nanocrystals, which are about 50 nanometers or so. And so we want to do something similar with looking at injectable nanocomposites with these magnetic materials, which could be used for, as you can imagine, magnetic muscles or actuators, where you have these magnetic nanoparticles dispersed in a polymer matrix or grown within the polymer matrix. So we've focused on MMS6, which is a mineralization protein which is found in those magnetosome membranes in the bacteria. Of course, if we just look at these magnetotactic bacteria and try to get the MMS6 proteins, we cannot really get enough to do all of our studies. So we end up cloning and producing these in E. coli so we can get enough quantities of this MMS6 protein and then attach it to our polymer templates and end up forming these crystals. So this summarizes some of the work that we've done in looking at these templates for biomineralization for these magnetic crystals without this MMS6 protein. If we want to do this, and this is done completely outside of the bacterium, of course, in vitro, we get very small particles, junk. And with these MMS6 proteins attached to the polymer templates, we end up forming these nice magnetite crystals, about 30 to 40 nanometer magnetite nanocrystals. And again, we've gotten rid of the organic phase so we can actually get good TEM images here. In addition to the full-length MMS6 protein, we've identified the C-terminus of the protein as the active part for the mineralization. So if we just use the last, or the C-terminus 25 amino acid peptide, instead of the full-length protein, we end up forming similar nanocrystals. We've also formed some manganese and iron oxide containing crystals. So, of course, living organisms, or like these magnetotactic bacteria, as well as some other species like homing pigeons and so on, produce magnetite nanocrystals. And one of the things we wanted to see is this limited to magnetite? Can we make related compounds with this that are not just magnetite? So we made manganese and iron oxide containing nanocrystals, which could have use in MRI imaging, as well as cobalt ferrite nanocrystals, which are now ferromagnetic instead of super paramagnetic. So you can see very well-defined platelet-like crystals that are formed with these proteins, even though in vivo there is no cobalt ferrite that's produced by any species that we know of. But the same protein, or actually the C25, can allow us to produce these very nice cobalt ferrite crystals as well. We've also looked at magnetic properties of these different composites or samples that we've formed, and this is sort of since I'm assuming there's not a whole lot of familiarity with magnetic measurements, this is sort of the test that we do. It's zero field cooling. We start off with a really cool sample, apply a magnetic field, and once this blocking temperature is reached, then beyond that, as you increase the temperature, thermal fluctuations dominate, and then you increase the temperature and then go back, and by doing that you can look at what's called remnant magnetization. And so if you look at the results for these different samples, on the left you have these magnetite nanocrystals that are formed in the presence of MMS6, and on the right we have cobalt ferrite, which is one of the other examples that I showed you. And then we have a bunch of different controls, including no protein, or even those synthesized or fabricated with ferritin, which is known to be an ion-binding protein. As opposed to the MMS6, we have a clear difference in the magnetic properties of the crystals fabricated with and without MMS6. Similarly, with the cobalt ferrite, with no protein, you see these really small crystals with very low blocking temperatures, which the high blocking temperatures and high magnetic irreversibility at high temperatures is indicative of much larger, well-defined crystals that are formed. And with the conjugated proteins, you get the largest crystals with the best magnetic properties. And one of the things that we're also doing right now is to look at the mechanism of action of MMS6. This protein, we don't have a crystal structure yet for the protein, and so one of the things that we're trying to do is to understand the mechanism of action and the ion binding activity. These are some X-ray fluorescence studies at a surface, the Langmuir-Blodgett trough. You can see that as, if you just have a buffer with iron chloride, the moment you add MMS6, which is itself an amphiphilic protein, and forms micelles on the surface, immediately you see a huge increase in the ion signal at the surface, which means that all the ion from the bulk is coming to the surface to bind with the MMS6. And so we've done some studies with some mutants of this protein with some random arrangement of the carboxyl and hydroxyl groups in these proteins, as well as shuffling and so on, different types of mutants. But in all these cases, it's the wild type that actually produces the magnetic response that we see. And with the mutants, we see very little response or ion binding. And so we are in the process of trying to elucidate the mechanism of action of these mineralization proteins in forming these nanocrystals. And one of the things that these polymer templates do in vivo in the bacterium, these are membrane proteins. And so these amphiphilic block copolymer templates also provide an amphiphilic environment for stabilizing these proteins so that they can function in a manner similar to that in the bacterium, because without the polymer, we don't see the kind of activity as well that we see with the protein and polymer. This is actually work that my collaborator Alex Travisett is doing on this project, and looking at coarse grain molecular dynamic simulations to understand how the presence of the inorganic phase actually can alter the assembly of the polymer with the protein, and that has been very useful for us in guiding our experimental studies. And I do want to put in a plug for his work with doing molecular dynamic simulations on graphics processor units, GPUs instead of CPUs, which allows him to do a whole lot more. And there's a WhoMD, which is available on the Iowa State or website. And as far as our future directions, we also through my sabbatical with David Tyrrell, we're also looking at some elastin mimetic peptides and some fusion proteins with making some fusion proteins of these elastin mimetic peptides with MMS6 with this leucine zipper pairs, so that you can have a reversible attachment of MMS6. So once you form these crystals, can you actually unzip this and try to have additional flexibility in the overall construct. These are also gel-forming proteins. Now we're looking at proteins instead of polymers, but it's a similar kind of system that we're looking at. So in summary, I'd like to just summarize the second part of the talk, where we've tried to make these biomimetic a biomimetic approach to making these nanocomposites, either with the hydroxyapatite or the magnetic nanocrystals. All of these, I should emphasize, there is no post-synthesis annealing needed to obtain these highly crystalline samples. Everything's at room temperature. We don't need to actually heat this to form these crystals. And so this templating method, and that in turn allows us to keep the polymer there, because if we have to heat it, the polymer is gone and it's no longer a nanocomposite. But this templating method allows us to control crystal size and shape, as well as placement of nanocrystals within this polymer matrix. So, summary, I'd like to thank everyone for their attention. I'd be happy to answer any questions that you have. And I found this on the web, so I thought this was very appropriate for this occasion. So I probably could have found a better picture, but this summarizes how I feel about Purdue, and I'd be happy to answer any questions at this point. So.
We'll take students' question first.
That's a nice tradition. I don't think it existed when I was here. Yes, thanks.
So, these nanocrystals, yes, you can make them, but what is the application of them?
Okay, so as I talked about many different systems here. For instance, the hydroxyapatite ones, we have this system now that we have this solution essentially which has the precursors to forming the nanocrystals and mixed in with the polymer templates. And one of the things that we're doing with our collaborators at the University of Iowa is to inject these into joints after trauma. So when you have trauma, you have cartilage damage, cracks in the cartilage. Of course, there are some chondrocytes that die right away, but they're looking at the zone surrounding that area of death where they have chondrocytes that can be rescued. And so if they mechanically stabilize it and also biochemically. So one of the other things we are also working on is to do some drug release from these nanocomposites, which would be the next step. But at this point, it's mechanical stabilization. These polymer gels themselves do not have mechanical properties that match that of cartilage. We're looking at a completely synthetic system here for just stabilizing something that as soon as the trauma patient comes in, something a surgeon can just shoot into the joint. And so this would be something they would shoot into the joint. It would conform to the shape of the defects because it's a liquid when you shoot it in and forms a gel instantly. And then the nanocrystals grow. And these nanocomposites, and I didn't show you the data, but they have properties very similar to that of cartilage. And so this really, and also it allows good bonding to the surrounding cartilage because these crystals are forming inside the body. These nanocrystals form a bridge between the cartilage and the material. And cartilage itself, when you think of it, yeah, it's, you don't think of nanocrystals when you think of cartilage, but there is actually the cartilage closest to the bone, which is mineralized, so it's not too different from that. And here we are talking about a small fraction of the crystals. So that's one example. Hopefully I've given a long answer, so I'll stop here. Yeah.
So for the geochemistry part of the talk, I have a question like what was causing the selectivity for the cancer cells? And so the transfection was like 10 to 15%. So how can it be improved further?
Yeah, that's a good question. So regarding the selectivity in cancer cells, of course, with polymers, there's a inherent selectivity because the cancer cells are faster dividing, and presumably it can get into the nucleus during cell division. That's been known, but it does not explain because some of the normal cell lines we had are also fast dividing, so that does not explain the selectivity of transfection. That's why we looked at the intracellular barriers, and essentially our conclusion was that the differences in lysosomal pH is what's causing the difference. And so what we're doing now that we know that, is to design polymers with different pKs that would better match that, to take full advantage of that, to improve that the transfection efficiency as well as selectivity of transfection. So really we are focusing on the polymer chemistry and the end blocks now, whether we use things like polydimethyl aminoethyl methacrylate, isopropyl, or other variations of these cationic blocks which can have different pKs, which can allow us to tune that better instead of looking at targeting ligands, which has been the normal method followed in literature to obtain selectivity of transfection in cancer cells versus non-cancer cells. So we think this is a new way to actually target cancer cells for gene delivery or even drug delivery. Yes.
It was interesting that the cationic micelles were valuable and non-toxic. I would expect the deuterionic and the anionic to be better than the cationic. How do you explain that?
Yes, yes, I completely agree. Actually, we did not. I didn't show you a comparison with anionic and zwitterionic. The cationic ones are known to be cytotoxic, but in this case, our saving grace is that we have this middle block, which is completely non-ionic. We have polyethylene oxide, polypropylene oxide, tri-block in the middle, and that's really what gives us the edge over Xgen, which is completely cationic. We have cationic blocks, but then we can adjust the ratio of cationic to a non-cationic part, which allows us to tune the, of course, it's a balance between good complexation and transfection efficiency versus biocompatibility, but I think we've found that sweet spot where we can get pretty good transfection efficiencies, but at the same time not allow it to be as cytotoxic as you see with, say, Xgen. But the anionic ones, we haven't, they're very, well, I didn't show you the biocompatibility data, but yes, you're right, they're much more biocompatible, and those are the ones that we're actually using for the other project with the hydroxyapatite biomineralization. They don't work so well for gene delivery because you want the cationic groups to help complex DNA or condensed DNA. The anionic wouldn't do it then. So, yeah.
What is also very interesting to me is that the commercial carriers, Xgen, did not show that much difference.
Mm-hmm.
So what is it that makes the pentablocks so unique in terms of producing selective gene transcription?
Well, actually, if you look at the data, the simple answer is that the pentablock copolymer is pretty bad at promoting endosomal escape. That's what it boils down to. And compared to Xgen, Xgen does a great job, and it's known that Xgen does a great job facilitating endosomal escape. And that's what's lacking with the pentablock copolymers. But that's an advantage for us because that's what I'm saying is true for normal cells, which have a low pH environment in the endolysosome. But in cancer cells, where the pH is much higher, it's easier to get out of there. I mean, with the pentablock copolymers, that's what it essentially boils down to. Yeah, yeah. So it's so it's essentially the pK of the polymer and the differences in pH in the two cell types. And so the reason we have the selectivity is because for this particular pentablock copolymer, it doesn't do a good job of facilitating endosomal escape in normal cells, which is good for us. Not that we planned that, so it turned out to be good. Yeah.
Small question. I believe you showed a phase diagram with different phases in the measure plot. So then I believe also you're doing molecular simulations.
Mm-hmm.
So how do molecular simulation predicted phase diagrams compare with the experimentally obtained phase diagrams?
That's a good question. Our collaborators have worked mainly with the pluronic copolymers as opposed to the pentablock with the polyelectrolyte, because it gets so much more complicated to do molecular dynamic simulations with polyelectrolytes and so on. And so they focus mainly on the pluronic, looking at the inorganic nanoparticles and so on. And for that, we get pretty good agreement between the phases that we predict from the molecular dynamic simulations versus what we see with our small angle X-ray scattering or neutron scattering studies. They have not done the polyelectrolyte one, so because it adds another layer of complexity to be able to do MD simulations with changing pH and so on. So, yeah. I don't know if they plan to do it, but yeah.
I have one question regarding the nanocomposite part.
Mm-hmm.
So you talk about the possibility of using the nanocomposite material as a replacement of cartilage.
Just wanted to clarify, that's not a long-term replacement. This is mainly as my colleagues in the med school have me understand, they just want something for 2 or 3 weeks. Right after the trauma, they want something to immediately stabilize that cartilage, prevent further chondrocyte death. After that, they don't care because there'll be some natural healing, and those population of chondrocytes at risk are rescued by then, and they don't die if mechanically stabilized. In addition, they've also want to release some antioxidants to also biochemically stabilize them. We haven't done that part yet with these nanocomposites, but.
So which type of metal materials are you planning to use?
For these, the polyacrylic acid pentablock copolymers do the best job in allowing us to get about 40% inorganic in there. So, and the crystals, and so those are the ones that we're focusing on.
Thank you. So, okay, let's thank Dr. Mallapragada for a wonderful talk.
Thank you. Thank you so much. Again, I'd like to say thank you. It's great to be back, and enjoy your time at Purdue. It's wonderful. I had a wonderful time, and I hope so do you.